A very good morning. I would like to present my project and a very good morning. I would like to present my project entitled Effects of 1,4-benzoquinone on DNA repair pathway and detoxification enzymes targeting hematopoietic stem or progenitor cells of different lineages. Benzene is categorized as most stable aromatic hydrocarbon, which is found in industrial solvent, and it has been categorized as common environmental contaminant. It has been found in the environmental in which humans are exposed to benzene through the automobile exhaust, cigarette smoke, and also gasoline vapors. Yes, benzene is toxic because it has been Group 1 in International Agency of Research Cancer, in which chronic exposure of benzene has been reported in bone marrow depression and many types of leukemia. And then it has been reported that benzene has to be metabolized in order to produce reactive metabolites known as phenol, catechol, hydroquinone, and benzoquinone, which play an important role in benzene toxicity towards the hematopoietic systems. Overall, the metabolism of benzene involves two pathways which happen in liver and also in bone marrow. In liver, they have metabolic activation to produce phenol, which are then metabolized by the MPO, also known as myeloperoxidase enzyme and NTO enzyme, to produce 1,4-benzoquinone and hydroquinone, which are very reactive, and it has been reported to induce hematotoxicity and leukogenicity. What is hematopoiesis? Hematopoiesis consists of a group of blood cells which are known as the progenitor cells, which actually proliferate and also differentiate in order to produce different types of mature blood cells. And the balance in hematopoiesis is very important in order to maintain a balanced system of production of mature blood cells, because if there's anything in regulation or any chemicals which actually can cause production of leukemic stem cells, also known as cancer blood stem cells. Actually, benzene hematotoxicity and leukogenicity have been reported to occur via multiple mechanisms which are involved: chromosome aberration, oxidative stress, alteration of gene expression, error-prone DNA repair, and also epigenetic regulations. DNA damage can play a fundamental role in the hematopoietic system function, because if there is the repair have occurred but is not repair, it can cause the stem cell to proliferate in order to produce stem cell containing the mutation. And if the DNA repair occur in an error-prone way, that means the misrepair occur. This also can cause mutation in the stem cells. Thus, it's very important to study the DNA repair system occurring in the hematopoietic system. Apart from that, the double-strand breaks that caused by the benzene actually cause the DNA repair, which is error-prone, which also play an important role in the benzene-induced hematotoxicity. This DNA repair is divided into two main groups, which are known as homologous recombination and non-homologous and joining repair. But according to the report previously, both actually are involved in the genomic instability. It is very vital for you to study the benzene toxicity in the hematopoietic system complex, because hematopoiesis consists of niche. It's a dynamic microenvironment consisting of various types of progenitor cells which play an important role in production of mature blood cells such as lymphoid, myeloid, and also progenitor cells. Apart from that, previous studies have reported there is a lineage direct toxicity of benzene, which actually occur differently according to the different type of progenitors, such as myeloid, lymphoid, and also erythroid. Thus, it's very important to look at this lineage directed strategy. The experimental design actually starts with the sacrifice of an adult male mouse in order to obtain the bone marrow cells, which is actually cultured for 24 hours in vitro, and it is exposed to various concentration of the 1,4-benzoquinone before it has gone through the colony forming unit assay to produce respective progenitors such as lymphoid progenitors, myeloid progenitors, and also erythroid progenitors. Okay, and then it is subjected to DNA repair profile and also detoxification profile, which involve DNA PKC enzymes RAD51 for the DNA repair profile and MPO and also NQO1 for detoxification profiles. The results shows that there is actually differential responses among the progenitors in which, for example, for non-homologous end joining users who reported to be an error prone actually happened most probably in the myeloid cells, as you can see it occur even starting from the lowest concentration of the 1,4-benzoquinone. For RAD51, which represent the homologous repair, it is not actually significant in erythroid, but it is shown significant in myeloid and pre-B, but it starts as lower as the fifth concentration of 1,4-benzoquinone. For detoxification profile, myeloperoxidase, this shows that this is one of the metabolic enzymes which play an important role in benzene toxicity. And from here, we can see that the myeloid play an important role as well. Okay, and then for NQ1, which plays a detoxification enzyme, it shows that myeloid happen to be the most sensitive for the reduction of the NQ1 enzyme. Therefore, the promotion of error-prone is actually very important in the genomic and stability via chromosomal deletion, insertion, or translocations. Apart from that, the MPO and Q1 also are very important in the BQ and HQ redox conversion of benzene toxicity. So in conclusion, we can conclude that 1,4-benzoquinone exposure causes error-prone DNA repair and reduced detoxification, which varies among the progenitors, and we can conclude that it is lineage dependent. Thank you.